Today we are going to discuss about the callus culture. What is callus? It is a simply undifferentiated mass of cells or the meristematic cells. So if we have to go to know how callus is formed, we can take any explant from the natural source, like take leaves, we can take root tips, we can take shoot tips, we can take floral parts, and we will be able to know their specific role. They are growing and getting differentiated under in vitro conditions. So the explant which we are using, we can grow these explant either on the semi-defined media or the defined media. So what is semi-defined media? Semi-defined media means we are using synthetic media is one component, and second is our supplement which we are taking from the natural source. We can use tomato juice, we can use orange juice, or coconut endosperm water we can use. So semi-defined media means one thing is defined to us, another is undefined to us. The media which is defined to us is also called as synthetic media, and another is the media which we are taking from the natural source. And we can grow callus simply directly onto the well-defined media, like we have studied White's media, B5 media, or the mostly used media is the Murashik-Skoog media. So for callus, there are mostly two processes occurring. First is the basic mechanism is like from simply explant, there is the dedifferentiation process. Then from dedifferentiation process, there is again the another mechanism which is called as redifferentiation. If we talk about the dedifferentiation process, this is simply when we have taken the explant, it will be having complex tissue. When this complex tissue is grown onto the nutrient media, they simply form the simple type of cells. Those cells are undifferentiated cells. We can call these as a meristematic cells. So explant से हमने क्या लिया फिर? Explant से हमें बना simply meristematic cells. Complex tissue से हमें simple tissue मिल गया. Then for the redifferentiation process means when this or the meristematic cells are converted into the differentiated mass of cells. This is the process called as the redifferentiation process. So to form the callus, there are basically three sequential processes which is occurring there. Like first is the induction, means things have to be induced in the explant. Then when things will get induced in the explant, how things will get induced? First, if we have taken an explant, we have placed this on the nutrient medium, there should initiation of chemicals which will be able to utilize this nutrient medium. Then the process is like there is the doubling of the nuclear material. Then from when nuclear material is doubled, then the cell division will occur. Then this is the second step for callus formation. First is induction. For induction, there is the initiation of metabolic processes which are able to secrete and form certain enzymes which will be able to initiate the cell division. Then second is the cell division. So when initiation occurs, then what happens? They form simply mass of unorganized cells, that is the meristematic cells. So induction, then cell division. Then the third important process is the differentiation process. When we are having mass of unorganized cells, that is the meristematic cell, they then with the help of growth regulators or growth promoters, they get differentiated into different organs. This process is also called as organogenesis. So what we will do, we take an explant, then the dedifferentiation process will occur. Then from the dedifferentiation process, we are able to have callus in our hand. Then from the callus, we are again getting redifferentiation, means complex things are from either root or shoot, and this whole process is called as a organogenesis. So if we will talk about what explants we can use for callus culture, it can be any variety. We can take any part of the plant. We can take part from angiosperms, gymnosperms. But in case of woody plants, these things are little tricky because we don't match our environmental conditions with the natural conditions where these woody plants exist. So for explant or for callus culture, we can use embryonic tissues, We can use like fleshy storage organs like seeds are there, aerial tubers, shoot buds, apical, even apical meristems, pith, root, segment of root or stem, any floral part, or even we can use inflorescence as well. Inflorescence, even it has been observed, if we will take a undetermined cells, if we will take a undetermined cell like parenchyma cells we are having, or cambium cells or embryonic tissue, they too have capacity to get differentiation. They too have capacity to get differentiation, that is they are able to form the callus. Or if we talk about the nature of the callus, what is the nature of the callus? This callus may be either sometimes compact in structure, or it may be loosely woven, that is friable in nature. There are different colors which we have observed while culturing our explant into the in vitro conditions. It may be green in color or greenish yellow tinges there, or sometimes we have seen, in many cases we have seen deep red colors for calluses. By keeping these callus under appropriate culture conditions and manipulation, we are able to go for organogenesis or embryogenesis as well. So what they have, when we have like scientists have started this tissue culture technique, earlier they used to do this tissue culture technique with the dicots only to initiate the callus formation, and these experiments were started in 1959, and these series of experiments were started in the 1959. Then starting from the dicots, then scientists shift to mosses, ferns, or even gymnosperms as well. So if we have to take the explant, we have to be very sure that these explants should be taken in a sterile conditions. So if we are taking our explant either from the natural source or either from the greenhouse, it will be obviously contaminated with the microorganisms. So before inoculating onto the nutrient medium, what we have to do, we have to go for the surface sterilization. We have to go for the surface sterilization, and this too we have to, so we are having different surface sterilizing agents. We have to choose our surface sterilizing agent which will be dependent on the sensitivity of the explant, Even sometimes we can use directly, like if we will take a bud which is covered by many floral leaves, we will remove these floral leaves under in vitro condition, under laminar airflow. So directly we can place the inside the bud directly onto the nutrient medium. We are having different chemicals which are using as a surface sterilizing agent, like we are having sodium hypochlorite, it's 2% we can use, or for sodium hypochlorite there is availability of commercial solutions, that is 2% volume by volume, or calcium hypochlorite we can use 9 to 10% weight by volume. This calcium chloride can be used for 15 to 30 minutes. Even I have discussed this thing that we can add certain liquid detergents to it, like T-Pol is there, Tween 20 or Tween 18, which are liquid detergents. We can add 1 drop of liquid detergent in these solutions, where we will be able to eradicate all the organisms which are present onto the surface of this explant. Even we can use woody shrubby plants for tough. We have seen that for tough plants like woody plants or shrubby or underground tubers, for their surface sterilization we are using 0.1% mercury chloride, and the exposure time is 10 to 12 minutes. So after exposing this to mercury chloride, then we go for washing with the sterile water 3 times. Even it has been observed that if we will take a parenchyma cells, simply parenchyma cells, these parenchyma cells are not able to form callus, or they are not able to initiate or induce callus formation. So even it has been seen, if we will take a single type of cells, they are not able to form callus, but this is mostly specified with the parenchyma cells. Or even if we take different cell lines or samples or explants where we are having vascular tissues, there is more formation or quick formation of calluses because they are having different types of cells present in it. Even whatever we are doing, it should be all done under aseptic conditions. Even they have observed that we are having many different explants, like they have used potato tubers as an explant for callus formation. Even they have used tobacco pith and tobacco pith where they were able to form the callus. Most of the work which scientists have worked or we know till now is because scientists have taken a model plant organism, that plant organ or model plant, that is tobacco. That is tobacco in case of plant tissue culture. So if we will talk about the nutrient media which is able to form the callus, as we have studied in the media formation, we should be having what? Macronutrients, micronutrients, iron should be there, carbon source should be there, vitamins, sometimes we add the plant extract as well, and plus the phytohormones which play important role in the organogenesis. We have to take, we can't take a one media for all the explants, so we have to be sure whatever the explant we are taking is suitable, or that medium are having all the requirements present in it which is suitable for the particular plant. And we have to keep this media under suitable favorable conditions, whether we have to keep this medium or our explant onto the agar liquid media or on in the liquid medium, or onto the agar means solid medium. हम लोग एक्सप्लांट को कहाँ पे ले सकते हैं? या तो सॉलिड मीडियम में भी ले सकते हैं या तो फिर लिक्विड मीडियम भी ले सकते हैं. Even we can, for it has been observed for callus formation we keep mostly our explant in the dark. Why we keep explant in the dark? The reason behind is that if we are keeping our explant which is cutted, so there will be some cells which will be half cutted, there will be some cells which are fully cutted, or sometimes some lesions will be occurring while cutting this explant which we have to place or inoculate onto the medium. So if we will keep this plant under sunlight or light, so what will happen? Photosynthesis will occur. So it has been observed that there is the formation of certain phenolic compounds. It has been observed that there are formation of certain phenolic compounds. That phenolic compounds leads to browning of our medium, and this browning will lead to the death of our explant. To reduce such conditions, we keep mostly our explant in the dark, where we have to be sure that we have to go for a callus formation. And we have to keep constant light, not directly light, diffused light we have to keep, and temperature up to 25 plus minus 2 degrees centigrade. Temperature is 25 plus minus 2 degrees centigrade. If we will take example, if we will take a segment of root, that root will be dedifferentiated and form the callus. Then from redifferentiation process will occur. Whole the process is able to process only when we know the nutritional requirement of that specific plant. जो ये redifferentiation process है, this is depends on the nutritional requirement of the plant, and it depends on the genetic makeup of the plant कि इसका जो genome है plant का वो क्या है, वो उस पर भी effect करता है. So when there is a formation of callus, we will be able to observe phenotypic changes. So we will, when we are keeping our explant onto the so for example onto the solid medium, when it induces cell division, obviously cells will be larger in size. So with the help of microscope, we will be able to see the change in the architectural pattern of this callus from explant into the callus. कि कैसे कैसे ये जो explant है ये कैसे कैसे change shape कर रहा है. And the shape obviously and the insertion of callus या callus induction के लिए क्या क्या चीज़ें होनी चाहिए या callus कैसे induce होता है, this depends on the plant genotype. This depend on the plant genotype first. सेकेंड कि प्लांट का ओरिजिन कहाँ से है, उसका फिजियोलॉजिकल स्टेटस क्या है, और हमने किस स्टेट में इस एक्सप्लांट को इन विट्रो कंडीशन में लाया है. इवन ये फैक्ट देयर आर मतलब लाइक वी आर हैविंग सम कैलसेस जो बहुत टफ होते हैं, कॉम्पैक्ट होते हैं, और देयर आर सम कैलसेस विच आर फ्राइबल, को लूजली वोन होते हैं. दिस फ्राइबिलिटी ऑफ कैलस इज डिसाइडेड और द नेचर ऑफ द कैलस इज डिसाइडेड बाय द जीनोटाइप, कि इसका जीनोम क्या है. और सेकंड द एज. अगर आप ओल्डर ज़्यादा लोगे, उसमें मोस्टली ऐसा देखा गया है कि जो ओल्डर मतलब एक्सप्लांट होता है, लाइक इन केस ऑफ इफ यू टेक द एक्सप्लांट फ्रॉम द वुडी ट्रीज, दे फॉर्म द कॉम्पैक्ट, मतलब टफ चितम बनाओ कॉम्पैक्ट में कैलसेस. या अगर हम लोग यंग लीव से लेते हैं, दे फॉर्म द फ्राइबल कैलस, एंड मोस्टली फ्राइबल कैलस आर प्रेफर्ड वन because of many importance. So even we can observe the phenotypic variations, like इच्छा tough callus, इच्छा smooth callus. तो उससे भी हम अंदाज़ा लगा सकते हैं कि हमने जो हमारा जो एज था एक्सप्लांट का वो क्या है. If we have taken the young tender explant, that obviously will lead to the formation of friable callus. So by this we will be able to also observe कि हमें जो हमारा एक्सप्लांट वो हमने कहाँ से क्या लिया था. लाइक इन केस ऑफ वी आर हैविंग वन, मतलब लाइक वी आर हैविंग डिफरेंट वेराइटीज़ फॉर ईच, लाइक वी आर हैविंग डिफरेंट वेराइटीज़ फॉर मेज. इवन इट हैज़ बीन ऑब्जर्व दैट वी आर हैविंग डिफरेंट वेराइटीज़ फॉर टमाटो. टमाटो की एक स्पीशी दूसरी स्पीशी से इतना वेरी करती है कि वी आर नॉट एबल टू गिव ए मतलब डाउन फ्लो की इसके बाद ये करेंगे, इस स्टेप के बाद ये करेंगे. वो आज तक अभी तक जनरल प्रोटोकॉल बना ही नहीं है टमाटो स्पीशीज़ के लिए. व्हाट आई विल से, टमाटो स्पीशीज़ आर हैविंग सच वैरायटी एंड देयर इज़ such a chromosomal instability for which we were not able to generalize the protocol for isolation and culturing under in vitro conditions. So if I will talk about the how we have to form the callus, or how we will be able to form the callus under in vitro condition, if we will make a diagram for this, this will be like that, like this. So yeah, how the diagram is visible on your screen. Is it visible on your screen? Okay, so if we have to see, I will take a highlighter. So if we have to see that if we will taking the explant, so what we will do with the explant, we will go for the surface sterilization of explant. From the surface sterilization, then we have to inoculate this explant onto the medium, onto the medium. Onto the medium like here. So when medium is there, we directly go for the organogenesis process. When simply we take an explant, we inoculate onto the liquid or solid medium, then with the help of growth regulators, we will be able to directly form root, or shoot. The process in which we are able to convert this callus into the root or shoot is called as organogenesis, and this organogenesis process is only because of addition of the phytohormones or the phytogrowth regulators adding to the medium. And we can go for stepwise process, that is we can take a explant onto the solid medium, then we will initiate callus formation, then from callus we will add auxin in higher concentration than the cytokinin. If oxygen is in higher concentration, what is going to happen? Root formation will take place. Then we will transfer this callus in with the root into the next medium, which will be having higher concentration of cytokinin with respect to the oxygen, will lead to the formation of shoots. So by taking explant onto the different media conditions, we will be able to form either or shoot in a stepwise process. So this is the how callus culture are formed inside the in vitro condition. When both root and shoots are formed, then we will take this plant for hardening. Simply from test tube to the pot, we will be able to allow it to form deep roots in the pot. Then from again we can use we can take this plant as an explant, then we will grow it under in vitro condition. This is how callus cultures are formed. Now if we'll talk about how when we have to go for the subculturing of this callus. When callus is growing inside the test tube or inside the funnel, or sorry, inside the beaker, or inside the, mostly we don't use beakers, or inside the flask. So what we do when it's growing, when it has reached 2 to 3 centimeter, then we take this explant and aseptically, then we cut it into either 4 pieces or into 8 pieces, then we go for subculturing of this. So but there is one problem while going for the subculturing of explant. What is happening there during the subculturing? There is sometimes variations are occurring, which leads to the variants like somocoloral variants. So mostly people are not, or the plant biotechnologists are not comfortable subculturing of the explant, because this variation leads to the histological changes, cytological differences, and these variations will lead to different strain and different cell lines. Sometimes these variations in these explant are also important for the crop improvement programs. If there is such variation occurring inside this callus or inside this explant, then we can go for screening of this explant, whether which important gene has been transferred, or what is, if there is mutation, which type of mutation is this, positive mutation which can be used for crop improvement, or if it is a negative mutation which is not beneficial for us. So if it is not beneficial for us, then we have to discard this explant. And the most important is the callus maintenance. So we keep callus continuously onto the medium, so we have to transfer this. After several weeks, we have to transfer this callus onto the new medium. So if we will keep callus onto the same medium, this will obviously declaration of the growth, necrosis, or the browning of the medium, and this will lead to the aging as well. But how this aging are processing, how this aging are occurring inside the callus, what is happening actually there? Aging may be due to the many reasons, like first will be the exhaustion of the nutrients. जब nutrients खत्म होते हैं तब भी aging हो जाती है, or sometimes nutrients are not able to diffuse to the explant, or sometimes there is evaporation of some nutrients, due to which there is accumulation of other nutrients present in the medium. Sometimes this may lead to the toxic effect, or other reasons will be the accumulation of secondary metabolites. As we know these secondary metabolites are not utilized by plant for itself, but these secondary metabolites are important for us. Due to these secondary metabolites, these may be toxic to the explant, due to which these explant are not able to grow. So what we do, mostly we take a small piece, 5 millimeter size, then we take this on to the 30 ml fresh medium, and the jar size should be 120 ml. जो हमारा बीकर है that should be 120 ml, और जो हम लोग उसमें मीडिया डाल रहे हैं वो कितना डाल रहे हैं, 30 ml, and the explant which we are taking is the 5 millimeters. This is how we are culturing, subculturing our eggs plant under in vitro conditions. Even it has been seen that by this way we are able to maintain cell lines up to 15 years, and this process has been observed in case of सोयाबीन मीन्स 15 साल तक हम लोगों ने इससे क्या किया इस प्रोसेस से एक सेल लाइन मेंटेन की है. इट मीन्स 15 सालों में इस प्रोसेस से उनमें उतनी वेरिएशन नहीं आई है. सो मोस्टली हम लोग क्या करते हैं, डिम लाइट में ही रखते हैं इनको, डिफ्यूज्ड लाइट में रखते हैं इनको 25 प्लस माइनस 2 डिग्री सेंटीग्रेड. If somewhere it is 24 written, so don't get confused. So callus storage is important. First what they did was they start preserving this callus at low temperature 5 to 10 degrees centigrade for 6 to 12 months. But later on they observed that there is the loss of this callus, so means this was not recommended. क्या किया उन्होंने, सिंपली पहले उन्होंने लो टेम्परेचर पे लिया at what 5 to 10 degree centigrade for 6 to 12 hours. लेकिन उसकी वजह से क्या हुआ, जो हमारा कैलस है वो पूरा लॉस हो गया. देन अनदर मतलब वन ऑफ द कंपनीज that is called as American Type Culture Collection that is located in the Rockville, Maryland. So they are having ability to maintain the callus cultures even from, but still the results are not that satisfactory. So whatever the cultures they are maintaining in their labs, they are able to exchange this material with the other labs as well. Even sometimes they go for mailing, like they send from one country to another country in a liquid nitrogen via the air services. So this is about the whole about the callus culture.